It's exciting to announce that our phase 1 portion of the study with Talquetamab was actually just published in the New England Journal. And at this year's ASH meeting, we presented the phase 2 portion. So, and I can get into a little bit of the differences, but basically Talquetamab is a novel first-in-class agent that's a bispecific antibody. And so it engages CD3 on the T cells and a novel target known as GPRC5D, which stands for G protein coupled receptor family C group 5 member D. So this protein is overexpressed on myeloma cells preferentially, even more in malignant than normal plasma cells, and also generally is not very highly expressed in other human tissues, with an exception perhaps of heavily keratinized tissue. So in the phase 1 study in New England, we found that this drug worked very well, even in advanced myeloma patients, response rate around 70%. And we had characterizing safety profile. And here at ASH, we updated both efficacy and safety at this meeting. So here, there were 3 major cohorts. The first was getting a dose of 0.4 milligrams per kilogram every week subcutaneously. The second was 0.8 milligrams per kilogram every 2 weeks. And the third cohort was either one of those doses, but specifically in patients who had had prior T-cell redirection therapy. I should mention that some of the patients from the phase 1 study were included in this data set, but the majority of these patients were only for the first time presented at ASH. And also I would distinguish this study because in the phase 1 study, we studied a variety of doses from very, very low to RP2D and higher. And we studied both IV and sub-Q formulations, but here in this study that's being presented is only subcutaneous. So the findings, to put into context, I think we just need to briefly review who the patients were. These were typically of relapsed refractory myeloma patients. There was a high representation of extramedullary disease, about 60%, be it by high-risk cytogenetics, ISS3, or extramedullary disease. And in particular, ISS3 extramedullary have been known to be high risk, even in the era of T-cell redirections, which is CAR-Ts and bispecifics. And also these patients, approximately 95% were progressing on their last line of therapy. And also, again, almost all patients are refractory to CD38 antibody, and about 3/4 were triple class refractory to PI, IMiDs, and CD38. So, in a heavily treated 5 lines of therapy over about 6 years. So in this population, we found that the response rate was 73 and 74% in the 2 cohorts respectively. And that response was maintained in triple class refractory, pentadrug refractory, ISS3, high-risk disease, pretty much all the subgroups, except perhaps extramedullary disease. And those patients did respond very well, but response rate was 50%. And also I would highlight that the time to response was quite rapid, around 1 month, and time to deepest response was actually also rapid, around 2 months. So that was the first 2 cohorts. And then I think the other finding that would be important would be the third cohort, which had a response rate of about 62% into prior T-cell redirection. We also did find some efficacy in terms of durability. We found that the PFS was about 8 months and 12 months in the 0.4 and 0.8 dosing respectively. And the median duration of response was 9.3 and 13 months. And for those patients who had CR or better, we didn't even have a median reach. So to summarize, I think what we find is that this is a highly active agent, both by response rate of around 60 to 70%, depending on the cohort, and also a deep and durable remissions. I think to kind of show where this drug might be heading, we should also look at briefly the efficacy. And so grade 3 and higher toxicities were uncommon, except hematologic, and those were less than 1/3 of our patients, and typically early on. Also infections, which have been plaguing some of the other bispecifics, particularly BCMA. We didn't see high grades of grade 3 infections, only about 10 to 15%, 2 COVID deaths, and none in the phase 1 portion of the study in New England compared to many more COVID deaths. And we see good COVID antibody response to vaccine, and also relatively low rates of IVIG. But we did have some unique toxicities with this agent, which included the CRS, which has been reported with all the bispecifics, about 70% low grade, But the unique things are rashes, which are quite manageable in dose early part of the cycle, nail changes, and loss of taste. But I bring up this safety in terms of future directions because we know that myeloma is very genomically and immunologically complex, and these initial single-arm studies with monotherapy is not how we always use the drug in the future. And I think because of the safety profile, this drug is readily combinable with other agents because there's lack of overlap. So it's being combined with all the standard myeloma backbone drugs. It's being combined with the other bispecific known as Talquetamab, and also with checkpoint inhibitors, and also confirmatory phase 3 studies going on with this agent in various combinations that's already accruing. And of course, there's great interest in moving these agents in earlier lines of therapy, but a lot of excitement about this novel MOA, novel target in myeloma.